Hello, welcome to my channel. This is Dr. Mukesh Nitin, and in today's lecture is lecture number 2 for Python. In today's lecture we will be discussing what is Python, the previous introduction. Now we will be discussing some new commands what are involved in Python, and I will start with gradual things, how Python can be installed, what are the that will be required. The most important primitive thing is that Python is already present in your Linux system as well as in Mac system. But if you want to download Python in your Windows, you can easily download Python in your Windows. I will share the link of Python downloading for Windows in our description in YouTube, where you can easily download the Python and you can start coding. In previous lecture also we have already discussed that we are using different kinds of text editors by which we can code for Python, like Notepad, Sublime, PyCharm, Spider and Google Colaboratory. In this lecture also we will be continuing with Google Colaboratory, and also I will be teaching you different kinds of codes that will help you to understand how Python works for a biological problems or for an IT problem. If an IT people is also watching this is a very good scope for them also, that they can check what is the scope of Python and coding for IT persons. While for biotechnologists we will be solving the biological problems, so being a biotechnologist you will also get a very good idea about how we have to code. For doing that I will be again going to the screen, but before that today we will be discussing what are the different types of data that we will be using for Python. And first thing it comes for data analysis in Python is string. What is string? String is a sequence of symbols that are coded in inverted commas or inverted colons. So there are different types of special patterns by which you use inverted commas in Python. So I will be teaching you in more detail about that. Also I will give you some interesting codes. So for that, what we have to do, first we have to go to the Google. You will see that I had already logged in with my Gmail. Similarly you will also log in with your Gmail. This was already mentioned in your previous lecture. Google Colab or Google Colaboratory. When we will be doing the Google Colaboratory, we will enter the welcome to Google Colaboratory. A new window will open for Google Colaboratory. Since we have different kinds of files here which I have made, but if you are joining as a newcomer, I will be starting again. I am giving you one more revision to that. So we will open a new notebook. Okay, we will go and open a new notebook. While opening the new notebook in Google Colaboratory, it will open, and here when the Google Colaboratory new notebook opens, you will go and you will rename that topic as Basic Python Training 2. Since I had already done one lecture where it was a Basic Python Training 1, this will be Basic Python Training 2. So here the first thing we will be discussing before starting this particular Google Colab, the first and foremost thing is that you have to connect it. Since you are a new one, so when you are connecting it to your system, it will be going to the option of connecting to, and it will connect directly to your Gmail. When it will directly connect to your Gmail, it will take some time and it will initialize and it will get connected to your email. So here once it is connected, you will see the option of RAM and disk is there. So those who are joining this second lecture, they will also get an idea how the coding works with Python. So here we will discuss what is string. Okay, the second foremost thing is how the string is being used in Python. What are the different methods of putting inverted commas, single or double. Okay, so for doing that I had already prepared some notes here that I will be guiding you, and I will be also attaching this to the mail to whom the notes will be shared according to that to your query. But before starting to that, I want to explain some important things for you. If you are working with Python in Notepad, Python is already installed in Linux also, and there are some options by which Python can easily be installed in your Linux. Mac is also having the Python, so if you want to code for your Notepad, you can gladly code for your Notepad. But for here, I am teaching you in Google Collaboratory, and if you want that how we can develop the software in Python through Notepad or through this one text editor, I will also arrange other classes for that. Now, when we are opening the Google Collaboratory, here the things are how we will be using this Python strings to the code. Okay, so if we write RNA set, okay, this is one variable, RNA sec1. Okay, I am making one variable, and we will use the string or the sequence in a single inverted comma. If I write in single inverted comma 80, okay, so when I do this, it will not give me any error. So we are starting to code again. So if we are making a variable name RNA sec1 and we are entering the data and the string in this, and we are putting it as single apostrophe, then there it doesn't return any kind of error. When I again use the variable, and this time I give double inverted comma, and run it, it will also doesn't give me any error, no return in error. But when I write another variable, I will give double inverted comma, then I will use the string or a sequence what we are making, and I make it a single inverted comma. You can see there is a red mark arrow that is indicating that there is something wrong. When I do this, it will return an error. So the first thing it signifies that when you are writing a string in inverted commas, it should be identical. If you are using, then you will be using single apostrophe opening and closing. When you are using double inverted comma, then you are using double inverted comma opening and closing. But you cannot use hybrid mode of double inverted comma opening and closing with single inverted comma. Also, you can use the single inverted comma within the double inverted comma, that is available. So I will make the correction again, and it will run. This is the first basic steps in Python. When we are using the string of a sequence, these are the different kinds of simple rules that we have to follow while doing the Python. Now, in order to open a new code, I will be using command Control MB together. When I use the command Control MB together, it opens a new window where you can again code. In the same code font, you will be getting more and more new code text editors. My next target is to make you understand how we will use small function in strings. Okay, so for doing small functions in a string, what we will do, I will make a variable RNA sec1, okay, and I will make a code, okay. This string which I made is a string variable with a symbol sequence where it is 8080G, and that is entered with RNA-sec variable having the values. The RNA-sec1 variable is a variable that is giving you or assigning you the values. Now, our motive is to change it to a lowercase. For doing that, I will be using another variable. I will be using another variable, say RNA low, okay. And in that, what I will do, I will call the RNA variable 1 dot lower function, okay, dot lower function. And by doing this, I have to since I had assigned the RNA-sec1.lower function to RNA low, I have to call that variable of RNA low to visualize the answer. When I do control plus enter together, we will get the result of string in lower case or in a smaller case. So this is also done. Now I will be using certain more codes for you, control MB, that will give you an idea for a biologist to use biocodings. As we are here training for biocoding, we should not get confused from biocoding with medical coding. Medical coders are generally medical transcriptionists. They do research in different kind of field. But in field of biocoders, you will be working in field of developing a Python software. You will be working biological problems, addressing the biological problems with Python software, or in software development, or in database management system, or in genomic analyst, or in data scientist, or biodata scientist. All these fields are included in biocoders. So now we will be again coding for a new biological problem. Here my problem is that I want to replace sequence where there is T should be replaced with U. Okay. So for doing that, I will be again giving a variable name, like same variable name I will use RNA-sec1. Okay. And here I will give the nucleotide sequence. Okay, I will give a nucleotide sequence A triple T, AT. This is a hypothetical sequence which I am giving. By using this hypothetical sequence, we will be working how to do the coding. For my problem is to replace that T with U. So now I want that I should replace my sequence into a RNA format. Okay. So let's do, in order to reduce confusion, I will use DNA. Okay, I will use, this is a DNA sequence. Okay. Now I will convert it into RNA sequence. Sequence 1, where I will be using DNA-sec1 dot replace command, where I will be replacing T with U. Okay. Again, it is also the same process. Here I have mentioned that I am replacing T with U. So in order to replace T with U, what I have to do, I made the command, replace command. Now I have to call my variable, and when I will do this, I will be able to replacing all the thymines with uracil. Okay, this is our second biological problem. So by doing the codes, you can understand that how biology being used by a Python programmer in order to solve the small problem. Later we will be developing into more complex problems. Now I will again go to control MB to a new command terminal, where my task will be to calculate the CG content. Okay, how to calculate the CG content? So my task is how to calculate the CG content in a sequence. So for that, we will do a trial basis for a small sequence, and later we will be using for a big sequence. So for doing for a trial for a small sequence, what we will do, we will write a variable name sec, and like this is a hypothetical sequence which I made for you in a variable. Now my task is to identify how many C and G content are present in this sequence. So first I will use to identify the variable. So I am using C variable in order to identify it. So what I will do, I will use RNA-sec. This is my variable. You can give it DNA-sec also, but I am using RNA-sec, depending as it's your choice. You can give another name also. I am using this RNA-seq1. Now I have to count. I have to count what I have to count. I have to count what is C content. Similarly, I will be using another variable where I will be using G as a variable where I will be counting G as a variable where I will be counting the same variable RNA-seq variable I will call, and I will use count function to count G content. Okay. Now I will use another variable total where I will count C plus G. Okay. Now I will call total variable to check how many C and G contents are there. So it shows that there are total 13 C and G contents present in this system. So we can calculate this. G content 1, 2, 3, 4, 5, 6, 7 is the G content. And what is C content? 1, 2, 3, 4, 5, 6. So total it is 13 contents present for G and C. So like this, we are able to calculate the content of GC content in our program. I will again use Ctrl MB to write a new command. Here, what my program will be, how to calculate the CG content in terms of percentage. Okay, now my task is to identify or count how many CG contents are there and what is the percentage of CG content present in that sequence of our query. So let's go to that same terminal and we will check in our command line screen that what I will do. I will use the same command RNA-seq CG content. I will use the same command and I will paste my same variable RNA-seq and the same CG content. But here the things have been changed. Here we are not calculating the CG content. We are calculating the percentage of CG content. So for doing that, I will be using the variable total is equal to C plus G. Then I have to use the command len because I have to calculate in the length of what? Length of our variable RNA-seq1. In length of our variable RNA-seq1 is the total sequence. So we are calculating the percentage of CG in the total length of our sequence in terms of percentage. So now I will again calling the variable total and we will get the lettered answer to this. So it is returning 48.14% is the total content of CG present in our sample. So I hope you are enjoying this biocoding with Python. So there is no problem. If you are a biologist also, you can code for Python. And if you are already in IT, you can also understand that what are the different kinds of nucleotide and DNA sequence, what are the hidden things. You can use biological problems to solve using your Python skills. So in both the cases, there is win-win situation because there are a lot of job scopes are available in this special field of biology, especially medical sciences and plant sciences and health sciences and agriculture. A lot of people and lot of research labs in India and abroad are looking for a bioinformatician or a biocoder or an IT person who has a little skill of about a biology or a little skill of biotechnology. Now I will try to show you another problem and let's see how we can solve the problem. So here the new problem is that in a given nucleotide sequence or in a sequence or in your biological query sequence, we are asking to find the specific position of codon. Okay, codons are you can say the nucleotide sequence codon is start codon, end codon. There are different kinds of codons where there is a composition of 3 nucleotide sequence together making in a codon. For example, we want to find a start codon AUG, okay, in our sequence. So our problem is how to find the position of AUG sequence. AUG, position of AUG sequence from our query sequence from start to end. Okay, so for this, what we have to do, we have to find this. So I will be sequence, query sequence, and I will be using again this command again one more time in another query, where I have to find this sequence, whether my position sequence is present or not. And this time what I will do, I will remove AUG position, but we want to find AUG position. So what in this query, what I did, I have a sequence where there is no AUG present, and I wanted to find AUG in that sequence. So when we are able to finding the AUG in that sequence, and when we do control enter, we get -1. So the answer -1 is because we don't have that particular sequence in our finding. And when we are trying to find the sequence query AUG, my task is to how to find the number of AUG from sequence from end to start to end. How to find the number of AUG sequence from our query sequence from start to end. In this, if we take our sequence, for example, I am taking the same code RNA-seq1 and giving this, and this time what I want to do, I want to extend this sequence a little big. Okay, now I want that, let's get the value RNA-seq1 dot, and we want to count a particular sequence AUG, okay, from our coding. So when we plan to do this, and we will check the control enter, you will get 4. So here what we are doing, we wanted to find the exact position of our codons present in a given sequence, where it is present and how much it is present in the given sequence. So we get the sequence 4 in this. So with this, you are getting the idea about how the string, you can use the Python coding program for your string to find the value in your value. I am ending the lecture session 2, and we will be learning some more interesting Python coding in the coming types, where we will be working more interestingly on knowing about different kinds of perspectives and angles of Python. So stay tuned with this channel. Don't forget to like, share and subscribe this channel, and more videos will be coming. So stay tuned to Dr. Mukesh Nitin channel. Have a nice day. Thank you.